Hi, this is Alex from devstories.eu. AlphaFold, Protein Structure Prediction, Episode 8 of 21. A single typo in a sequence of thousands of amino acids can be the difference between health and severe genetic disease. But when you sequence a patient's genome and find a novel mutation, how do you know if it is the root cause or just harmless biological noise? AlphaMissense provides a mathematical answer to that exact problem. A missense variant is a genetic mutation where a single DNA letter change results in substituting one amino acid for another in the final protein. The average human genome contains thousands of these variants. Many are entirely benign. They alter a peripheral part of the protein without affecting its overall function. Others disrupt the protein entirely, leading to serious genetic conditions. Sorting the dangerous variants from the harmless ones is a massive bottleneck in clinical genetics. AlphaMissense is a model specifically built to classify these effects. We need to clear up a common misconception right away. AlphaMissense does not predict the 3D structural changes caused by a mutation. It does not output a new file showing a deformed protein shape. Instead, it leverages the structural and evolutionary context already learned by the AlphaFold network to calculate a probability. It outputs a continuous pathogenicity score ranging from 0 to 1. This score represents the likelihood that a specific mutation causes disease. Take a concrete scenario. You are analyzing a patient's genome and you isolate a novel single amino acid substitution in a critical gene. A valine has replaced an alanine at position 142. The patient is ill, but this exact mutation has never been documented in medical literature. You need to know if this substitution is pathogenic. You query the AlphaMissense database for this specific variant. Under the hood, the model evaluates the evolutionary conservation of that exact position by looking at multiple sequence alignments. It checks if nature has tolerated changes at this position across millions of years of evolution. It also evaluates the structural context. An amino acid packed tightly into the hydrophobic core of the protein is much more sensitive to changes than one floating loosely on the exterior surface. AlphaMissense processes these evolutionary and structural signals to return your score. A score close to 0 means the variant is likely benign. The protein probably folds and functions normally. A score close to 1 flags it as likely pathogenic. The substitution almost certainly breaks the protein's stability or function. To make this data actionable, the model applies predefined thresholds. Scores falling above the upper threshold are likely pathogenic. Scores below the lower threshold are likely benign. Anything falling in the middle band is classified as ambiguous. This classification allows you to instantly filter genomic noise. Instead of designing expensive, time-consuming lab experiments to test every unknown variant in the patient's genome, you immediately focus your clinical resources on the few mutations AlphaMissense flags with a high score. Here is the key insight. The true impact of this tool is its scale. AlphaMissense does not wait for queries. It has already calculated the pathogenicity score for all 71 million possible single amino acid substitutions across the entire human proteome, cataloging the results before a patient ever walks into a clinic. If you enjoy the podcast and want to help keep the show going, you can support us by searching for DevStories EU on Patreon. That is all for this one. Thanks for listening and keep building.